Okay, chapter 14, section 2, autotrophic pathways. Okay, autotrophs, remember, these are the organisms that can take carbon dioxide gas from the environment, fix it into carbon skeletons, carbon chains that can then be utilized in the formation of polysaccharides, or I'm sorry, polysugars, saccharides, sugars, amino acids, whatever. So, going to be looking at quite a few microbes here, fall into this autotrophic here. This is going to be things that also, because remember, these are going to be the organisms that are phototrophic, okay, and things that are chemolithotrophs. They utilize organic compounds, chemo, litho, they make them themselves though. So they take in carbon dioxide, fix it themselves. The CO2 is what is utilized for the biosynthesis of all organic molecules. The amino acids, the nucleotides, the lipids, and the carbohydrates. Mixotrophs, these are ones that are bit hetero, bit auto, depending upon the environment they're in, depending upon the state of the environment they're in. They can go one way, but then snap back, boom, boom, depending upon changes. They are not all one or the other. Now remember the Calvin cycle, which many of you probably when you think Calvin cycle, if you remember it at all, because it's probably been a while. Intro, cell, molecular cell biology, plant biology. Notice there, I said plant biology. You think plants when you see this Calvin cycle, because it's how plants take the ATPs and the excited electrons from the light cycle, and they use it to power the Calvin cycle, the fixing of the carbon dioxide into 3 carbon chains, which can then ultimately be used to make fructose or glucose. Well, it turns out microbes have been doing it, but doing it longer. It's not a complete copy of the Calvin cycle that you've learned in plants. It's a modified, a rudimentary version of it, because remember, they have been here longer than we have. Going to see this, versions of this in the purple bacteria. The cyanobacteria, the purple bacteria, definitely plants. But the version, if you go and you look at the enzyme reactions, you look at the different protein complexes between the cyano, the purple, and the plants, no, they're not all going to be the same. Overall themes, yes. Homologous reactions, orthologous reactions, orthologous proteins, enzymes, probably. The key here is Robisco. Robisco reduces oxygen to glyceraldehyde 3-phosphate, G3P. Hopefully you remember G3P. G3P is one of the 3 carbon chains you get after you split fructose 1,6-bisphosphate. Okay? Remember, you get G3P and dihydroxyacetone phosphate, DHAP. G3P is the part that goes through the second half of glycolysis to become pyruvate. Calvin cycle needs 12 excited electrons, 18 ATPs to synthesize 1 fructose 6-phosphate from CO2. It is very energy dependent. Because it is very energy dependent, in the making of all the enzymes and having the Robisco around, could be a bit of an issue. Have it now, make it all you need, then get rid of everything, remake it, because now is the time. Oh no, you just missed it. No. So, what many of these microorganisms have done is evolved to have what are called carboxysomes. Okay, these are proteinaceous microcompartments. Okay, proteinaceous, means that they are not lipid-bound vesicles like you would find in a eukaryotic cell. Okay, it's not an organelle, it's not a vesicle, because remember that's a lipid bilayer. Here, it's going to be a proteinaceous covering that's encapsulating a region of the cell cytoplasm. And in that cytoplasm, you're going to get a lot of the stuff you need for Calvin cycle, primarily Rubisco. Got to protect it because Rubisco is highly reactive in the presence of O2. O2 competes for CO2 when it comes to some of the enzymatic reactions that you'll find in these cells. Rubisco is one of those reactions. It's interesting, Calvin cycle predates oxygen. Got to remember, early Earth was not the Garden of Eden we have now. No, the environment was hot. The environment would kill you within minutes of the heat. The gases of the environment were more methane, ammonia, and things like that. So, carbon dioxide levels were way beyond any trace amounts of O2 that may have been there. So those first organisms, those first cells, would not have been aerobic. They would have been anaerobic. They would have also been looking to other mechanisms to make their own organic molecules. Fixing of CO2 would have been a good thing. When you're looking here, you see these are carboxysomes, okay? These are not lipid-bound vesicles. These are not lipid-bound organelles. These are a collection of proteins with a little bit of the cytoplasm contents in there, and it's all bound by this protein meshwork, holding everything in place. In many of these microorganisms, the Calvin cycle per se is not going to be, the three-stage event you learned in Intro Cell and Malac, or all the different steps involved if you took plant biology. Here it's more often than not referred to as a reverse citric acid cycle or reductive TCA. Keeping in mind that all of the enzymatic reactions of the citric acid cycle are reversible. So instead of taking the, all the oxaloacetate, adding the pyruvate, and then going through all those steps to, decarboxylate, remove carbon, release it as CO2, stripping off those excited electrons, you're going to reverse the process. Start with oxaloacetate and go backwards, the fumarate and all of that. Fumarate, malate, alpha-ketoglutarate, and all that, so that you end with citrate, which can then be modified, and you can get back, through via, gluconeogenesis, you can get to a 5-carbon or 6-carbon sugar. Turns out it's actually more efficient than the Calvin cycle. Yes, it's going to require more electrons, 24 hydrogens, but only 10 ATP. In the process of reversing the citric acid cycle, instead of releasing CO2, you're fixing CO2. Taking gaseous CO2 out of the environment, fixing, and moving on. There are some enzymes, as that last bullet said, some steps in the citric acid cycle are not quite reversible. So you're going to have special enzymes, alpha-ketoglutarate synthase. Pretty much wherever you've lopped off a CO2, you're going to need a different enzyme to add the CO2 back. One of the steps substrate level phosphorylation to make ATP, or actually it's GTP, but then that third phosphate gets transferred to ATP. So we usually just skip ahead and just say ATP. Well, that's another irreversible step. So you'll have to have a different enzyme. But anyways, you start oxaloacetate. Reduce it, you get malate. Malate, twist, tweak, you have fumarate. Reduce it, succinate, succinate. You add in a coenzyme A. There's one of those steps. So you have to also, this is very energy driven, so you're going to have ATP, add a carbon dioxide, add a carbon dioxide. Now you have citrate. Go through, do a little release of the oxaloacetate, add on a CoA, you now have acetyl-CoA. Wait a minute, acetyl-CoA is only 2 carbons. Well, add on another one, pyruvate synthase. And now you are gluconeogenesis, glyceraldehyde 3-phosphate, G3P. The G3P, or any of these other steps throughout here, can be utilized elsewhere. You know, as amino acids, alpha-ketoglutarate can become glutamate, can become glutarate, and all sorts of stuff. Or, you know, comes glutamate, and then a whole handful of the pyruvate, add on an amino group, and this, that, and the other, and you have another handful of different amino acids. So, this is how some of those microorganisms can fix carbon dioxide and come up with pretty much anything they need for their cellular metabolism. Other pathways, there are alternate mechanisms for CO2 fixation. We're not going to go through these. All these green non-sulfur bacteria utilize what is called a 3-hydroxypropionate bicycle to fix CO2 into pyruvate. Yeah, I've read about that once. Not going to try to lecture on it. Archaea also utilize a version of this. They fix bicarbonate and or CO2, bicarbonate HCO3. It can be fixed as, you know, small short chains, 2 carbons, 2 carbons, 2 carbons, 2 carbons. You link a bunch of those 2 carbons together, you can make pretty much anything. Reductive acetyl coenzyme A, reductive acetyl CoA pathway. So basically, you do whatever it takes here for these to get acetyl CoA. You're going to fix CO2, 1, 2, 3. And you're going to skip the whole reverse citric acid cycle, whatever. You're just going to take enzymes and just literally power through. It's more efficient, but the problem is all those different steps, as I just mentioned on the last slide, also give you means of using them elsewhere. You know, you take the citric acid cycle intermediates or the reverse citric acid cycle intermediates, and with a twist, a tweak, a cut, and a paste, and you have pretty much any amino acid you could need in the cell. You know, so if your amino acid pools get low, you can take any of those and replenish. Once those are up and everything's good, you just start feeding the overproduced amino acids back in. You know, cut, paste, twist, tweak, and now they're any of those intermediates. Gluconeogenesis, you take those carbon sugars, you know, 6 carbon, go through the shunt, you end with the 5 carbon pentose for nucleotides. You can do all these different things with them. This mechanism doesn't allow for that. This is straight up, we're going to go 3 CO2s, or 3 carbon, 3 carbon, 3 carbon. Brute force, but yes, it saves you energy, but it doesn't give you any wiggle room for usage elsewhere.